So now let's take a look at what these functional groups are. I can't emphasize it enough. You have to learn the functional groups. Can't be any more explicit than that. You need to be able to identify them when they're present. You need to be able to describe their properties. And you need to be able to name the molecules that contain these functional groups. When you go to organic chemistry, I told you it's a weeder class. A lot of people fail out of organic. I don't want that to happen to you. If you have a good baseline knowledge of what the functional groups are, their properties, how to identify them, you're going to have a step ahead of your classmates. So this is extremely important. Sit down and learn the functional groups. I'm not going to spend a lot of time, just point out some special properties here. Again, you can do this on your own. First group here, the only nitrogen containing functional group we'll learn about, that's the amino group. You could see here signified by NH2. Now what's special about the amino group is that it acts as a base. Now do you recall what a base is? Can you define it for me? That's right, a base is a substance that reduces the concentration of H+ in solution. Now what is it about the NH2 that makes it a good base? Well, remember, we have 3 unpaired electrons in nitrogen because it has 5 valence electrons. But we also have an unshared pair that's not pictured. So here we have the amino group. So now in the presence of a proton, that proton can share that lone pair, now creating an NH3 group that is positively charged because we have an additional proton that has been added to the structure. So an amino group can be uncharged or unionized, or we have a functional group that can be ionized. What type of bonds can the ionized form of the amino group participate in? That's right, they can participate in ionic bonds. I'm going to start to call them electrostatic interactions. We also know that by nature these bonds are polar. So an amino group can be polar and charged at the same time. That's going to make that end of the molecule that contains the amino group reactive. It's also going to make it hydrophilic. Where will we see amino groups? They are very abundant in amino acids, hence the name amino. So look out for that. Next up, a carbonyl group. I'm going to circle the functional group here. C double bond O is a carbonyl. Now again, that's polar. Oxygen's going to be partially negative, carbon partially positive. So that's going to change the reactivity of a hydrocarbon when I substitute then a carbonyl. Now there are 2 different types of carbonyls based on their position. The first is going to be called, the molecule is going to be called a ketone. So a ketone refers to the molecule which contains the carbonyl. The reason this is a ketone is because the carbon is sandwiched between 2 other groups, typically carbons. So it'll be carbon, carbon, carbon with an O, double bond O attached. We're going to see examples of that when we look at steroids, an example of a fat there, a lipid, steroid lipid. Now an aldehyde is different, also carbohydrates. Oh, we got to put that on there. Carbohydrates can be called ketoses. Os means sugar. The C double bond O, the carbonyl, is in the ketone form. The second version is the aldehyde. Now, it jumps out at you right away. In an aldehyde, one of the groups attached to the carbonyl is a hydrogen, making that the terminal end. Does that make sense, right? You can't build any more off that hydrogen. So aldehydes tend to then, the carbonyl tends to be towards the end of the molecule. Again, it's polar. Those sugars that contain an aldehyde will be called aldoses. So the difference between glucose and fructose, for example, or the difference between certain sugars, are going to be based on the position of the carbonyl. Next one, this entire group I'm circling, is a carboxyl group. Do not call this a carbonyl and focus here. We always have to look at what's attached to the C double bond O. In this case, when you have an OH group, the entire thing is called carboxyl. Now we know this is highly polar, right? We have a partial negative here with the oxygen. We have a partial positive here with that hydrogen. So this is going to be very hydrophilic. We're going to find these in amino acids and fatty acids. Notice the commonality here. Carboxyl groups are acidic. Now what makes an acid special? Can you define what an acid is? That's right, an acid donates H+. Well, now let's look at this functional group here. What I hope jumps out at you, man, we have these two oxygens in very close proximity from an atomic standpoint, atomic level standpoint. Man, they're highly electronegative. So even though we have a covalent bond here between the oxygen and the hydrogen, that in many cases is short-lived. That oxygen is more interested and creates such a force that oxygen cannot withstand that it dissociates. That the oxygen rips away the lone electron of hydrogen, leaving a simple and lonely proton. Well, isn't that what the definition of an acid is? An acid is something that donates H+ to solution. So we can see that the carboxyl group can exist in two forms, either the unionized form here with a hydrogen attached, or the ionized form here. Well, we know when you add a charge, you're changing the chemical reactivity. We know that the carboxyl group can participate in electrostatic interactions, most likely with an amino group. We're going to see this as a common theme in protein structure. So carboxyl groups and amino groups are the two ionizable functional groups that are able to accept or donate protons and therefore can exist as either positively charged cations, this is the case amino group, or negatively charged anions. Last one here, hydroxyl group. Again, is polar or non-polar, that OH? Oh, good, you're catching on, that's polar. Right, so you add a hydroxyl group, you're increasing the solubility of that particular molecule. You're creating a functional group here that can participate in dehydration reactions that you learned about in the last chapter, the loss of water to create a bond. Steroids have hydroxyl, alcohol, carbohydrates, we'll see specific amino acids contain hydroxyl groups as well. So again, take home, identify them, know the special chemical properties, and then learn which molecules will possess which functional groups. Here are some other functional groups that are a little bit more specialized, only because most of them don't contain carbon. Here, we finish here with a carbon-containing methyl group. Methyl group, not very special, right, because it's non-polar. You'll see them in DNA, you'll see them in specific amino acids and proteins, and you'll also see them in carbohydrates. The last three are all non-carbon based. They are moving down, if you think about moving down a row on the periodic table. Here we have phosphate groups. PO4, you see the two negative charges. We find phosphate groups in DNA, RNA. You see them in nucleotides like ATP. We can attach phosphates to amino acids to regulate protein activity. The addition or removal of a phosphate group is an essential regulatory step in cell communication, for example. And so phosphates are often involved in regulatory reactions and also involved in the transfer of energy from one molecule to another. So these are often transferable functional groups. Sulfates, we don't see sulfates as often, but we do see them, SO4-. We can attach these to carbs, proteins, and lipids, again, as a way to change the structure or to change the function of those molecules. Last one we're going to see is the sulfhydryl group, a simple SH group. Very specific amino acid called cysteine contains sulfhydryls. We can see these sulfhydryls actually participating, adjacent ones participating in a bond called a disulfide linkage, which is going to be very important in stabilizing protein structure. So I can't emphasize it enough. Students struggle. I don't know why to learn these. I know you had to learn all the parts of the body. There's no reason you can't learn the different functional groups. So now let's try a poll. When ready, press pause, go pollev.com. Molecules within the same functional group have similar chemical properties, true or false?